Welcome to another episode of I Am Nano.
Putting the I in I Am Nano. I am your host, Irfani.
And putting the M, I am your other host, Monica.
And today we will be talking about conductive polymers to replace peripheral nerves.
Oh, that's exciting. Let's get into it.
So, in our body, we have a system called the peripheral nervous system that is composed of a network of nerves and motors that send signals from our brain and the rest of our body. It's a very important network to have. They send messages from your brain and spinal cord to the rest of your body, helping you do things such as sensing that your feet are cold and moving your muscles so you can actually walk.
Oh, nice. So if you get into an intense fight and you experience deep cuts or severe bruising, could you lose feeling in your arms and legs then?
Yeah, that can potentially happen. In fact, in severe cases, the peripheral nerve may have to be replaced. And right now, the general approach is to conduct autologous nerve transplantation, where the nerve from the individual is taken from another part of the body. But this can present multiple issues, including the shortage of tissue and the need to do multiple surgeries.
Oh, wow. That sounds so risky. But if I'm losing feeling in my arms and legs, I would take that option. But are there any other alternative methods to do this?
Many scientists are definitely exploring alternative methods in the field of tissue engineering. They are looking to use synthetic and biocompatible materials to develop molds to grow cells onto. Of particular interest is repairing the nerves that exceed 10 millimeters.
Oh, wow. So basically, making an artificial nerve.
Yes, definitely. So now, in today's lit update, we'll be discussing about a conductive hydrogel recently developed by Dong and colleagues, published in ACS Nano back in October 2020.
All right, let's unpack that. To start, what is a hydrogel?
Good question. I think we talked about this before, but just to recap, hydrogels is a network of polymers, which you can think of them as noodles that are cross-linked like a stack of fishnets. Now, these hydrogels are made of polymers that are water-loving, that allows them to absorb water. And here, Dong and colleagues use two different types of polymers, one called polyaniline, also known as PANI, and polyacrylamide, also known as PAM. And the resulting polymer is a microporous sponge that can allow easy exchange of materials and have enough surface area for cells to attach if they are needed. Now, how can we use this polymer to make the artificial nerve?
Well, they have to have good conductivity, right? Because they need to conduct electrical signals. And then, well, they must be stretchable because your body parts move around, right?
Right, yeah, that's true. So for conductivity, PAM does have some conductive properties, and in this hydrogel, since it is doped with the PANI polymer, it's approximately 400 to 500 times less resistive than the other reported conductive hydrogels. And even after they were stretched out to 2 times their length, they still maintain good conductivity. Another aspect that helps to enhance their electrical property is that the PANI polymer's ability to promote electrical signal conduction when exposed to near infrared light, and the current can increase up to 60%.
Oh, wow. But why is it important that the current increases by 60%?
So the nerve cells can propagate signal very quickly thanks to the sleeves of fat that surrounds the nerve. So the hydrogel's ability to increase its conductivity further ensures that the hydrogel can replace damaged nerves.
Oh, gotcha. So, have they tested the conductive polymer's ability to act as a nerve then?
Yes. Before testing it on real life animal subject, they have to make sure that it works outside of that system, which we usually refer to as an ex vivo study. And here, they took a muscle and nerve from a hop-toad's leg and then compared the resulting muscle tension and contraction produced by an intact nerve and one with a damaged nerve when a potential was applied. Both the intact muscle and one with the conductive hydrogel generated similar maximum muscle tension, and the time it took for the nerve to cause muscle contraction was also comparable between the two systems. And before they tested it on a rat model, they also confirmed that even after stretching the hydrogel 1.3 times the original length, 90% of the muscle contraction tension was still retained.
So, what is muscle tension? I've heard of it before, but don't actually know the definition.
Well, I think most people are familiar with it by calling it muscle stiffness. Many of us have experienced it before, especially, if we haven't worked out in a while. And I know that I have experienced it a lot on my shoulder and my back. It's really bad.
Yeah, so much pain from sitting at a desk by a computer all day these
Yeah.
days, eh?
Yeah. So they conducted a nerve injury mimicking surgery and excised 10 millimeters of the sciatic nerve on a rodent model and then inserted the conductive hydrogel. They measured the compound muscle action potential, which is the electric potential of a bundle of motor neurons caused by the activation of a bundle of nerves to evaluate the muscle contraction, so the muscle stiffness. And the resulting potential with the conductive hydrogel was like the healthy nerves, even after 4 weeks, 1 month after operation.
Oh, wow. So, a lot of animals went into this, from toads to rats. Interesting perspective. Any human subjects yet? Are they at the stage or still preliminary?
Not yet. They're still working out the details, but still very neat use of hydrogels, and the finding seems quite promising.
Oh, yeah, great research and very lit. I look forward to what the group does next. Link in the description for more details.
Yeah, for sure. And that's all the nano for today. Take care.
And stay curious.